All right, so we have started this week off with RNA. We introduced RNA. Tuesday, I lectured about transcription. Yesterday, we practiced. And now we're going to finish this off and we are going to lecture on translation. Now, translation is the final step in protein synthesis. In transcription, which is what you should have finished yesterday's practice activity, you have 2 attempts, so please try your best. Transcription is when we went from DNA to mRNA. And because DNA is trapped in the nucleus, transcription takes place in the nucleus, right? From DNA to mRNA, the most significant change in our base pairing rule is that we eliminated T and now we pair A with U, C, and G. This is essentially a recap of what I just said. Now, the most distinct thing that we're going to notice is that this process of translation, when we're going from mRNA to a protein, is going to take place at the ribosome. And the ribosome is located in the cytoplasm. And the one thing that I always say that helps me remember who does what is that ribosomes are the lean, mean protein making machines. And another thing that is going to continue to come up for this lecture is going to be the mention of amino acids, because amino acids are the building blocks or the monomer of protein. So I had kind of previewed this for y'all and I wrote it in your 1-page notes for transcription off to the bottom left in orange, I believe. But these are all going to be thrown around and we should be familiar with them and be okay with them and not let that overwhelm us, right? So here is a depiction of what's going on. This orange circle is the nucleus. Inside of it lives DNA. DNA cannot exit the nucleus. It is trapped there forever. So with the help of RNA polymerase, which is an enzyme, mRNA is able to go in and transcribe or copy, right, the code. Again, base pairing rules are used here. And the most significant difference is that A now pairs with U, C still pairs with G. We take this mRNA and it floats through the cytoplasm. So there's the cytoplasm, right? All this blue is the cytoplasm. mRNA floats until it reaches a ribosome. Once it reaches the ribosome, it attaches and we start to code for proteins. We're going to get into translation. So that's what we're lecturing on today. Now, translation is the process of actually making a protein. Making is synthesis, right? To make. And so protein synthesis, we're making proteins. We're making proteins out of the mRNA code that we took from the nucleus. Translation, again, is going to be located at the ribosome, and the ribosomes are free floating in the cytoplasm. Now, it is important that you note that there's 3 types of RNA, and I lectured about that on Monday. There's mRNA, which is your messenger, tRNA, which is your transfer, and then there's rRNA, which is your ribosomal. rRNA is responsible for mass producing ribosomes. And again, ribosomes are important because that's where proteins are made. Proteins are important because proteins code for our genes, which make our traits. So your eye color, hair color, height, etc. Now, first step, and I always think of this guy, your rRNA and your ribosome, they're attached to one another. It looks like Toad from Mario Kart to me. I don't know why, but I can't ever get that out of my head. So here, this bottom part, that's your ribosomal RNA and it's mass producing ribosomes. This top little circle is going to be your ribosome. Now the mRNA is going to attach to the ribosome and we are going to begin at our start codon. AUG methionine is going to be your start codon. The tRNAs are going to come in and they're going to bind to your codons. And again, I keep saying codon and I assume that you guys know what that means because that was a part of your vocabulary. But essentially that's going to be how we are going to read mRNA in groups of 3s called codons. So 3 nitrogen bases. The tRNA is going to have the corresponding anticodon. And again, everything fits together perfectly like a puzzle piece. So I have my mRNA sequence that I took from the nucleus and my groups of 3 are going to come in and plug the anticodons in. As the anticodons are plugged in, an amino acid, which is at the top of the tRNA, is going to be released. The ribosomes are going to create these bonds between these amino acids. and we're going to call these bonds peptide bonds, right? Peptide refers to proteins. We've mentioned this multiple times this year. I even told you about peptidoglycan, which was in the cell wall, and we talked about how archaebacteria and eubacteria, and we made that as one of the characteristics. And essentially all that we want to know is whether it has this protein in the cell wall or not. So ribosomes form these peptide bonds. We're going to call this amino acid sequence that's been bound together. It always looks like a little string of pearls to me. The polypeptide chain. Poly means many, peptide refers to protein. So once this chain is released, this polypeptide is going to be your protein. These tRNAs float away, they go get another amino acid, and they'll come in and plug in wherever they are needed. The ribosome is going to continue to move along and connect to these peptide bonds or these amino acids via peptide bonds until it reaches a stop codon. The stop codon is going to allow it to release the chain of amino acids, and essentially we've now created that protein. The protein is going to get folded and so that it can be used. So let's look at this to see if we're able to have a visual to everything I just said.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so there's another animation that we're going to watch here, but now let's talk about these codons, because as much as I would love to assume that everyone did their vocab and that you guys know exactly what I'm talking about, I need to just cover all my bases, right? So in order, how is the order of the bases on an mRNA strand translated into a protein? Well, it's going to be read in these groups of 3s, right? The biggest misconception that my kids have is when any numbers are involved, right? So a codon is going to be this 3-letter word, right? So it's going to have 3 nitrogen bases. One codon gives you one amino acid. Kids will make the huge mistake of saying that there's 3 codons to one amino acid. A codon is a group of 3, all right? So 1 codon gives you 1 amino acid. I always ask that question, and I'm almost positive it's in tomorrow's lab. 1 codon, 1 amino acid. How many nitrogen bases in 1 codon? 3. So as we're reading our mRNA sequence, we are going to input these codons into a codon chart or circle. If we take the 4 letters, A, U, C, G, and we make them into 3-letter combinations, there's actually 64 possibilities. There are 20 amino acids that help us code for proteins. And so you're not going to be asked to memorize any of them. The only one you probably will memorize is AUG because that is your start codon, and majority of proteins begin with AUG, which is your methionine. Now, I'm going to show you how to read both, and I've placed both in today's folder because it's going to help you as you work through the rest of the activities. Here is the codon chart. It's going to be a left, top, right, LTR. The way that we use this codon chart is we're going to, and this, if you're a visual person, you're going to ignore this slide because it's just going to be a bunch of words being thrown at you that mean absolutely nothing. But since we're given 3 letters at a time in a codon, the first letter is on the left, the second letter is on the top, the third letter is on the right. So essentially, if we're using this in application, AGU, AGU, where they all meet is going to give you your amino acid. So here, AGU gives me serine. ACG is going to give me, I say threonine. I don't know how you say it. Tomato is one of those things. Most of the time with amino acid sequences, you only write the first 3 or 4 letters anyways. So it's completely fine. No one is going to look at you funny if you say any of these wrong or right. It doesn't matter. So again, left, top, right, where they all meet is going to give you your amino acid. Now, the beautiful thing about these codon charts is that half of the time, the first 2 letters will give you the amino acid because it doesn't really matter where the third base corresponds because all 4 possible combinations, or all 4 possible combinations are going to give you the same one. I would still check and double check because sometimes you'll get these funny combinations where if you mess up the last letter, you can mess up by putting a premature stop or putting the wrong amino acid. And we'll talk about more when we make these mistakes when I lecture on mutations next week. So this is what the practice will look like. You have DNA. This is why there's T's in your original sequence. We're going to transcribe it. So we're going to convert DNA to mRNA. We're going to change all of our T's to U's. TAC, and again, I'm going to read these in groups of 3, right? Because that's how my codons are read. I have a lot of dyslexic tendencies. I've said this multiple times. So what I do when I'm actually handwriting or I'm coding for a protein, I always come in and I make myself these divides because this way my dyslexic tendencies are not going to get the best of me. Now, as I go through, TAC now becomes AUG. TGG is going to be ACC. CCT is going to give me GGA. And then last but not least, TCG is going to give me AGC. You're then going to take these mRNA codons and you're going to put them into a codon chart so that you can figure out which amino acids, right? So AUG is going to give you methionine, which is your start codon. Let me go ahead and pause my share so that I can show y'all the codon charts that I've put in y'all's. So these charts have been placed inside of today's folder. And so you'll have these available for you to use. And I put that note at the bottom. Codon charts are only meant to translate mRNA. Now, as you go through, ACC is going to give you threonine, GGA is going to give you glycine, and AGC is going to give you serine. Let's kind of wrap it up with this video. I think this video has really good illustrations because they're really Mickey Mouse. There's no like confusion. I feel like the more three-dimensional and the more accurate that the illustrations are, the more confusing they are for us. We are visual learners who like things oversimplified because the easier the better. Come on.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So this small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then the first tRNA can detach and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first three letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column. And the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
Okay, so here is one illustration. Like I said, the more three-dimensional and accurate that they are, the less I personally like them. I like them super simplified, but they're going to show us a variety of different depictions and illustrations. So we just need to make ourselves familiar with what to look for, because majority of the answers are in the labels. If you see a circle colored structure, that's almost always the nucleus. So you have to assume that DNA, and you can tell there it's a double helix, right? The DNA and mRNA. So this is usually illustrating transcription. If you see a nucleus, if you see DNA, if you see a single strand, almost always a picture of transcription. When we see this single strand and we are outside of the circle, that's because we're at the cytoplasm. This guy doesn't look like toad anymore, so I don't really care for it. But the ribosome and the ribosomal RNA are there, and the mRNA attaches. This little string of pearls, like I told you, that's going to be your protein. That's your polypeptide. Poly means many, peptide means protein, right? That polypeptide chain is your protein that was created. Now, we've seen this before. Again, DNA to mRNA, that is going to be transcription. We're changing the T's to U's, and that C, I always think of copy. Because DNA is trapped in the nucleus, this takes place in the nucleus. mRNA to protein. Trans means change. We're changing RNA into amino acids, right? And because the amoeba sister said that C comes before L in the alphabet, transcription happens before translation. Since proteins are made at the ribosome and ribosomes are located in the cytoplasm, those are always going to be two appropriate responses for the location of protein synthesis. Now here's another depiction essentially of what's going on. You're giving your DNA strand, right? How do you know it's DNA? Hello, there's a T right here. Transcription is when we go from DNA to mRNA. So we take all of our complementary base pairs, but we replace all of the T's with U's. We then take this mRNA and we are going to now read it in groups of 3. That's called your codon. Your codons are going to be input into the codon chart / circle, whichever one you want to use, that whatever works for you. And we are going to then get our amino acid from that group of 3. You're going to continue to code for groups of 3 until you hit a stop sequence, and now you've completed protein synthesis. You've made a protein. Let me go ahead and share this codon chart because I want to show you at least how to read them both. Again, I have a lot of dyslexic tendencies. I was never diagnosed, but I'm almost positive that I have some sort of dyslexia, right? So when I get charts like this, it doesn't help that I wear glasses, right? I've literally worn glasses my whole life. I was 5 when they put glasses on my face, right? We all know I'm like 16, so I've been wearing them for a long time, right? So here is the deal. I always take some sort of highlighter, pencil, color of some sorts, and I draw lines for myself. If this works for you and it keeps you from making a careless mistake, please do so. Now, almost always the first example that they're going to have you look for is AUG. And like she said, I had never heard it, but I think it's so cute. AUG, school starts in August, so that's where we start making proteins. That would be enough to help me remember. AUG. So where they all meet is here, and that's your methionine. That's your start codon, right? So that's how you would use the chart. If you are looking at the circle, here's how we read the circle. A, so your first base is in the middle, and then you're going to work your way out of the circle. A, U, G, methionine. Use whichever one works best for you. You will have a lab assigned to you tomorrow where you're going to be asked to code for a protein. So you're going to have to take your mRNA that you transcribed and put it into a codon chart. These codon charts only work for mRNA. Okay. How do we